Hi everybody, welcome to the Data Pre-Processing in Python workshop. This is workshop number 4 in the AIMS AI in Medicine curriculum, and this was supposed to be delivered live on March 10th, but unfortunately about half an hour before I came down with a very serious migraine. So if any of you wonderful, smart, and talented people want to use your new AI skills to combat migraines in some way, like predicting the time to onset of a migraine or figuring out the best drugs or the best combination of therapies to work with migraines, I think that is a very noble pursuit, and I think anyone who gets migraines would agree. So without further ado, let's get started. Okay, so obviously the times are not going to be relevant anymore because this is now not live. But the parts 1 and 2 will still apply. So part 1, I'm going to talk about where data comes from, what data pre-processing is, and also kind of give a high-level overview of some of the concepts seen in the demo. And in part 2, we're going to be jumping over to a Google COLAB notebook that I wrote. I'm going to talk about loading in the data, data exploration and visualization, different types of transformations that you can apply to the data, as well as data as machine learning input and kind of the format that the data needs to be in order to work with a machine learning model. Okay, if you had attended my Google Colab workshop, this slide probably looks fairly familiar. My name is Sarah Davis. I am one of the co-presidents of the AI Medicine Student Society this year. I'm currently a master's student studying medical AI and computing science at the U of A, but my background is in biological sciences and medical sciences. That's a lot of what I studied. And I've worked with just about every type of biomedical data out there. That's probably not entirely true, but it feels like that sometimes. So I will be kind of trying to teach some of my experiences with different medical data types to you guys. So the question, where does medical data come from, can be approached in 2 ways. So you can either talk about the source, so this is where the data is initially generated. Clinical trials is one example, which is published about in medical journals. Hospital records is one, genetic registries, such as those used for genetic counseling, or even privately owned repositories like from 23andMe, they send out the tests. Wearables, like your Fitbit, and others. Insurance providers hold a lot of health information. And recently it's been shown that even social media contains a lot of information that is very important to somebody's mental health. So there's been research predicting depression through tweets, etc. So that is starting to come into the limelight a little bit. The second approach is where the data is stored and like I have here, where the data is housed for access. So this can be public, so you can just log on to some website or even just visit some website and you can just download a data set right away. There is a lot of medical data specifically that requires proposals and ethics reviews, and the access is quite restricted, and that is just to make sure that there's no abuse of the information and the information is safe and not likely to be leaked. But specifically, I wanted to talk about where you can find toy data. That's good for learning. I have a few things here. Some is specific to health data, some is not. So Google has a search bar, a search tool called Google Dataset Search, and all of this can just be found by Googling as well. Where you can search for different data sets. Kaggle is a great place where you can not only see the data and download the data, but you can see notebooks of other people's interpretation of how the data can be used, which is really great for learning. The World Health Organization has a Global Health Observatory data repository, and so a lot of information there is available for free download, and that's great for health data. And then there is also the machine learning repository from UCI. I think that's UCI, not UCL. I'm not entirely sure exactly which university that is, but I've been to this site many times. Okay, so data pre-processing, kind of on a high level, is the manipulation, transformation, joining, filtering, splitting, and finalization of data for the purpose of some analysis. So this often requires an exploratory phase where you do data visualization and you really get a sense of what the data is and what its shortcomings are. That is important to know because the different treatments you will apply to it really has to be specifically tailored to the data that you have. So some common considerations that are important when you are doing this exploratory phase, you should be looking at missing values, potentially outliers as well. Those are less common. Missing values are very common. Normalization and scaling, just to make sure, I guess, your models have the best chance at learning like a robust representation and don't need as much training. Class imbalance is another one. So if you're predicting like tumor malignancy, if you have 900 like benign biopsies and 100 malignant biopsies, then in that case, if your model predicted that all tumors were benign, then it would get 90% accuracy, which is something that we have to look out for. That's a common pitfall. We're also going to look at the feature to example ratio, so that's the number of columns to the number of rows. The different data types and the most appropriate data types for different like columns. And additionally, deduplication. So if there is data that appears in like multiple columns, say, or there's duplicated rows, or there's been tables that have been joined and have created some kind of duplication, we want to have our data be in the most simple and non-redundant form possible. So that's another consideration. Okay, so here now I'm going to go through some important concepts, and most of these will be appearing in the code that I have prepared. I just think it's important to go through this just to get like kind of a primer of it, because it's kind of hard to explain through code sometimes. Okay, so first I want to talk about data shapes. So all of the data sets that we're going to be dealing with today comes in the form of a 2D matrix. So this is basically what one might think of when someone thinks about a data set. The form is rows and columns. So here in this example, we have 7 rows, including the 0th row here, and then 5 attributes or columns. So that's rows, columns, or examples, features. So when you read a data set, so say from a comma-separated value file into Python, into your Python environment using the library Pandas, and some other libraries as well, this matrix is called a data frame. And a data frame is a way to hold these 2D matrices, but also hold a lot of meta information about it, such as like the row numbers and the column names. Whereas if you were to use a more simple representation, like say a nested list of lists, it's less intuitive and less easy to keep that kind of meta information. You can also access something like the shape easily by calling the method .shape, which we'll see. And so it's a very useful tool for when you want to be kind of doing this data science, data slicing and dicing. So one column or row, so we could view each of those as a list if we wanted to, is called a series. So a data frame is normally made up of many series. And then if you have data in the form of this 2-dimensional matrix in this data frame, this is very often called tabular data. And this is just saying that it's flat, it's probably pretty easy to work with. Okay, so here's an example of data types. And so this is something that will inevitably come up in your data science journey. So we have strings. So an example of this would be name. This is just a sequence of any kind of character. Integer, so whole numbers. So an example of this is, say, the number of children that a subject has. Floating point, which is a number with some kind of decimal or the possibility of a decimal following it. The floating point, like I said. An example of this is systolic blood pressure. And then the last one that I want to talk about is the Boolean. And so this is kind of like a categorical variable. And you would normally see a condition as this feature name. So you could say the condition is visit emergent. So if visit emergent equals true, in this case, that means that this subject's visit to the hospital was like through the ER, for example. And if it's false, that means that this person was admitted through some other way. Okay, so there's also other data formats rather than the nice tabular data set. So one of them is image data. This definitely is something that is beyond the scope of this particular workshop, but it is a very fascinating field, and some people spend their whole lives on, machine learning on image data, or at least if they haven't already, they absolutely can. Common algorithms are convolutional neural networks that work well. I just have examples of some health-related image data. There's also longitudinal data, which is basically any collection of data points that is captured over time. So like your Fitbit heart rate, for example, all your Fitbit information is longitudinal because they take that information basically at every second of every day. And so the longer you have it, the longer the amount of information you have is. Brain waves, survival information, how long someone has survived for, and then also these, like an electronic health record is an example of longitudinal data. I don't think this star is supposed to be here anymore. I was originally going to talk a bit about longitudinal data, but I realized that with everything else that I wanted to talk about in this workshop, it probably wasn't going to happen. So maybe in future. I would love to talk. This is a lot of what I work on, so I'd love to talk about it in the future. Okay, so now we have two data sets, and say that dataset A and dataset B have a column in common. So in this case, the column is ID. So ID is just a number, one specific number here. But sometimes there is information in A and information in B that we want to consider as part of the same data set. So in this case, what we would do is we would do an inner join. So this means that whoever appears in both dataset A and dataset B will end up in the final dataset. And based on ID, they concatenate the columns in both of the datasets. So in this case, dataset B contains weight information, dataset A contains height. We have patients people with IDs 5 and 7. Finally, after this inner join, they appear with the height and weight information. So name is the same for both of them, so that is carried over but not changed. So another way to do a join is an outer join, where we do want to keep all of the patients or people that appear in both of these data sets, but not all of them have information for both height and weight. So if we're okay with some missing values, we can do an outer join, and that means that all of the IDs here encompass all of the IDs seen in both of these data sets. But because of the missing information, we're going to have these null values. And sometimes that's preferable. It's very much dependent on what you eventually want to do. Okay, so something else that we may want to talk about is one-hot encoding. So often we have categorical variables, and these are very, very common, especially in health data. But unfortunately, the machine doesn't know how to interpret these. So the machine wants numbers. We can give it numbers by doing this one-hot encoding. So say we have one column called pet. This is a string column. So we can actually replace this one column with 4, where we take all of the unique values that appear, and each unique value gets a column, let's say. So now, for the row, say the row that had pet cat, now within this list of 4, it will get a 1 under the cat heading if it has a cat, and a 0 anywhere else. Same with the dog, so 0 for cat this time and 1 for dog, blah, 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 all the way down. And then if cat appears again, it would get this one here. So this is great. Machines love this. Even though it's a little bit less nice for humans, this is just a necessary step. So this must be done for all categorical data before the machine learning part is done on it. Okay, and this is the last slide I'll quickly go over. What does the average machine learning model need? So first of all, we want a train, test, and potentially validation split, or we want to do cross-validation, but I'm not going to talk about that this time. So for each of the train, test, and validation, train is normally 80%. If you have validation, it's 10%, and then the test would be 10%. If you don't have validation, then the test would probably be 20. This is, I think this was explained in previous lectures, but there's a lot of information online about that if you want to know more. And then so for each train, test, and potentially validation set, you would need a feature matrix X, which is all of the attributes that you will use to predict what we want to predict. And then we need the target attribute Y. And so these need to be the same shape for the train, test, and val, except obviously the number of rows will be different. A machine learning model needs numeric values. So you can't, like I said before, you can't have strings, you can't have categorical. Boolean's easy to deal with because you just replace true with 1 and false with 0. We also, most of the time, need no missing values. So we need to perform certain types of treatments to, either drop the rows with missing values or to impute the values by inserting kind of some unoffensive value that is not going to impact the performance very much. We also need fixed width inputs, so that's what I was saying with longitudinal data. It's very difficult to compress all of that longitudinal information into a fixed width vector, but we need to do it. And then also another thing is we need if we've chosen a machine learning model, we need data that the model can explain. So this was something else that was covered in previous lectures and potentially workshops. If your model is too simple, it will underfit. If your model is too complex, there's the potential for it overfitting. And so I have down here, even if you can't fix it, you're responsible for it. So you are likely to be given data in the future, and if you go down the data science path, and then you are going to have to find a model that is a good fit for the data. And so that's a whole, field of study in and of itself. But with practice, that becomes a little bit more intuitive. Okay, so let's get to the demo, and I'll demonstrate everything that we need to do to get everything running. And, because you can pause this, please do follow along if you're interested. And while I'm going through the code, pause it, test your own hypotheses out, and fiddle around, see what all of the code does. Sorry, as with last time, everything is available on our link tree. So let me just check, make sure this is all good. Yes, okay, great. So you can see my entire screen now, and I will be following the tutorial that is found on this link. So this is what the link should look like. Here we have the slides, which I just went off of, the link to the Google COLAB notebook, and additionally the data and the tutorial. So that is on this GitHub page, Ames's GitHub page right here. So I am going to be following this README to get through this whole thing. And so in future, if you don't want to rewatch any of this, please do just follow these steps here. Okay, so first of all, we need somewhere to keep the data and the code. So we are going to want to open our drive and create a folder in it called Ames Data Workshop. So I'm just going to copy this here. Yes, okay, so this is my alternative Google Drive, so it's pretty empty. Here's the folder for the previous workshop. I'm going to create a new folder, and I'm going to paste the name that we are supposed to give it here, and in just a moment, yeah, there it is. Okay, so now we have this place to house the data. Okay, so that's great. Now our next step is to download the data. So I have made 4 data sets available in this repository, and some of them work together, some of them don't. And so what we are going to want to do is click download here, and you are going to want to download zip. If you're comfortable with Git, you can absolutely clone this if you would like, but for now we're just going to download the zip file. I have this easy to remember place where I keep it, So what we're going to want to do is we're going to want to download this zip file, wait for it to finish. And if we show in folder, we should see this zip file here. So we're going to want to extract. That seems fine. All right. And within here, we should have these 4 datasets. Varying sizes, as you can see, and so this is like just barely small enough to be able to be uploaded to GitHub, but I think, you know, everything should be okay. And of course, if you run into technical problems at all during this, just send me an email. That's absolutely no problem. So once we have that extracted, we want to go into our Google Drive, enter our AIMS data workshop, and we want to navigate to where we were in our desktop, and then we want to select all of the data sets that we downloaded and upload them. So overall, this shouldn't take too terribly long. The genetic data set that I have is definitely the biggest one, but I mean, if your internet's okay, it really shouldn't be too bad. Okay, great. We have this step done now. Hopefully that was very easy and painless. So our next step is to visit this link with the COLAB, and then we are going to want to save a copy of it, because this right here that we're seeing now is the master copy, and hopefully not just anyone can edit this. Yeah, changes will not be saved. So the idea, save a copy in Drive. I think, yeah, that should work. It'll create this copy. I like to locate it in the drive and move it from where it wants to live into where it should live, in my opinion. So if we do move to, we can move it to the AIMS data workshop. And we can also rename it so it doesn't have that copy in the title, because it's just nicer that way. Okay, awesome. So now, if we refresh, yeah, there it is. So great, we can close down the main copy. I like to do that just to not get confused. Okay, we did that, and it looks like now we are ready to go. So let's open this up. We select Google Colaboratory to open it with. I'm going to move this around. So now we should be absolutely good to go, and we can start with the demonstration. Okay, so first what we're going to want to do is connect our Google Drive, and this was talked about beforehand. So if you want more information about connecting your Google Drive and what it does and how to navigate this area, we do have a workshop about that. So first of all, I'm going to run this. I'm going to import this drive here so that we can mount my Google Drive in particular so I can access the data. You'll get this URL, just visit it, and continue through, allow, whatever. Everything's on your Google Drive anyway. You're not giving them any new permissions. And then enter the authentication code, the authorization code. Then it should spin a little bit and have mounted your Google Drive. So now, if we check this out, we should have our G drive right here so that we can navigate if we would like. And so now we want to navigate to our correct folder just by using this change directory command. So I'm going to run that as well. So looks like we're in the right place, but just to check, let's use the LL command. Yeah, and so here we have all of our data as well as our notebook. Okay, so now I guess as we can see in the table of contents, we are going to be getting into the data pre-processing now. So I have 3 case studies prepared to kind of show off some of the really important steps in data pre-processing. And so I will then get into case study 1, but first we should import our libraries. Again, this is all covered before, and these should be all of the libraries that we're going to need for today, so not too bad. Let those import. Great. Okay, so case study 1 is using the breast tumor data example. And again, if you went to the other Google COLAB workshop that I gave, you would have seen this. But we're hopefully going to try to kind of use it in some new ways that you would not have seen in the previous case. But I think it's just such a nice data set. So we're going to talk about here data loading, manual row and column engineering, as well as some feature selection. So there's a lot to know about loading in data. And just to give you kind of an idea of how much there is to kind of understand, here are all of the potential arguments you can give for the read CSV. You're not going to need a lot of them. I've selected a couple of important ones that you might want to look at. So if you want to figure out what sep does as an argument and nrows does, I would definitely suggest doing that. So let's just load in our data. So first of all, we're going to do this with only the argument passed in as the name of the file. So it should be able to just look this up, no problem, because we have this here. We saw that we're in the right place. So let's do pandas.readcsv and then the name of the file. So that went quickly. If we look at the first 5 rows, we notice we get this unnamed 0 column, which basically means that the dataset does not supply a column name for this first series here. And this series is the index of the rows, so it doesn't contain any real information. It was just saved with this file as a way to number the rows. And so what Pandas does when you load it in is it creates its new one. It wasn't sure that you wanted, if you wanted to use this as the index or not. So what we're going to do is instead, and I find I have to play around with this a lot to make the data look like I want it to, we're going to specify index call is equal to 0. We're going to do this all over again. And there's the nice data set. So we have our index and we have all of our column names. So the first thing that I always do is I check the shape. So it looks like we have 569 rows and 31 columns. This is a fairly small data set. I guess cancer data sets are quite small a lot of the time, but you know, this one in particular, it's at least easy to learn from and work with. So another thing is that you can perform an operation called transpose. And basically this just flips all of the rows and columns. So we have rows here, columns here. The transpose would take the columns, put them as the rows, and vice versa. So then you can see the shape would be 31 rows and 569 columns. This is helpful just when you're kind of working through the data, just to, you know, it'll either be easier to see or it's necessary for joining or something like that. So that's something I thought was worth mentioning. So like I had said before, the data frame contains a lot of meta information. So in order to access, say, the index, you can just type the data frame name .index. And in this case, because we didn't update the DF breast cancer, the transpose was not kept. So we didn't go DF breast cancer is equal to DF breast cancer.transpose. So none of this information was saved. And so to look at the index, we would just see use the .index. If we wanted to make a list out of the index, which I find I do a lot, you use this toList function here. And that just returns a regular Python list with no funny formatting or anything. Pardon me. Then we have .columns, which allows us to access the columns. So if I run this, oops, went too high. DF breast cancer.columns gives us back this index data type full of all of the column names. So if we wanted to update the columns to some other list of the same length, if we had defined some list somewhere, you can just set df breast cancer.columns as some other list. I'm not going to do that right now because I think these attribute names are quite informative for now. So I think one of the first things that is really important to learn is how to slice and dice. So this is how do we access specific subsets of rows and columns. And I'm going to show you two functions here, .lock and .ilock. So .lock is short for like .location. And so once you have a data frame, you can use this .lock with this particular format here with these square brackets. You can give before the comma the names of the rows that you want to keep or you want to select. You can also give the names of columns that you want to select. And this will return a new data frame that only contains the rows and columns that you want. So in this example here, this colon means that you want everything that the data frame has. So instead of specifying every row number, you just give this colon and it just gives you everything. So in this case, we want the mean radius. So this would return a series, just a list. I'm going to run this. And here it has this index here. And if you wanted to get rid of the index, you would use that dot to list that I talked about before. And then it just returns basically a list of these mean radius measurements of length 569 for each row. If you wanted to specify multiple columns, you absolutely could. You would just need a list within where the column should be specified. So we have list of mean radius and mean area. So if I run that, it actually returns a data frame rather than a series. So this looks a lot more like this than what the previous one did. You could also use the column, or not the column, the colon in the middle here to specify that you want every column in between these two. So if I do that again, this starts at mean radius and ends at mean area, but contains all of the columns in between, which is very, very useful. Because of what I mentioned before, I think, maybe you guys could imagine what this would return. It is just the entire data frame. So here we specify that we want all of the rows and all of the columns. That just gives us exactly what we had before back. So let's do a few examples with rows as well. And because rows are indexed by numbers, we can use these numbers here. So if we want row 0, we can run this, and this will give the columns and the values for each row. So this is a series as well, and to get rid of the names, you would just do .toList. We can do the same thing where we specify a certain number of indices within a list, and that'll return those, in this case, these 3 particular rows, and then all the columns, obviously. And we can also call it using this index and this columns. And again, it's, you might be able to guess what this whole thing would look like. This is exactly equivalent to this because it just uses everything that's available. Okay, we're going to get rid of that. There is also ilock, which I'll go over very quickly. So ilock is basically a way to specify location based on the position. So to call this, you would do dataframe.ilock, and then you would specify the positions of the rows you want and the positions of columns you want. So imagine then for this, we want to specify, here's a trick, if you put a minus, it starts at the other side. So instead of the first column in this case, this would give us the last column. So let's run this. And so this is the target attribute actually. So this, if you remember, is whether a tumor is malignant or benign. So that using that -1 is a way to specify the last element. We can do what we did before by giving a list. So this will give the 2nd and the 5th column. So that ends up being perimeter and compactness. Again, if you use this colon in between, this gives you everything in between those two positions. And then, once again, using the colons, we'll just, for both of them, we'll return the entire data frame. And you can do this again with the rows rather than just the columns. You can specify a list, and that will give you the first, fifth, and 100th rows with all of the columns. And then additionally, you can do the same thing with rows as you can with columns here, and just run it. And there you go, you get 4, 5, and 6. So I would say if you are a beginner here, learning these is a great way to start. Okay, so there's a reason that I taught you lock and iLock first, and that's because that is the kind of best practices way of specifying rows and columns. There is a shorthand where, say if you wanted to view a column, and the same is not the case with rows, but most of the time you wouldn't need to access rows like that anyway. You can specify the name of the data frame and give these square brackets, and then within quotations add the column name. So this is another way to view a column in a similar way. The problem is that often you want to select the target and you want to assign it to something else. And so you can't assign this method of accessing the column to anything else. It won't let you. That was like depreciated quite a while ago, deprecated. And so I would say avoid this if you're ever trying to change the data frame. If you just want to look at it, then it's fine. But if you want to change the data frame at all, avoid this. So here's a way that we can exclude rows and columns that we don't want kind of in a different way than this lock and iLock. So say we wanted to only select tumors that had a mean smoothness score that was above 0.1. And I assume that that would be more benign tumors than malignant ones, but I'm not a cancer expert. So first what we want to do is create a mask. So right here, this mask is being assigned to all of the values that come under mean smoothness that is greater than 0.1. And so this doesn't select automatically. I'll show you what mask looks like. So mask is actually a series of the length of the number of rows, and it returns true if the mean smoothness of that row is greater than 0.1, and false if it's less than or equal to 0.1. And so this ends up being a really, really powerful tool because Boolean indexing is very fast, especially when you have very large data sets. And so now we have this mask. We can actually use this mask to select using our location indexer here. So we can say we want the DF, we want the location where the mask is true. And so just by saying mask, that's the same thing as saying mask equals to true. So then if we do that, then we have selected 216 out of the 569 rows. And these are only the rows that have the smoothness greater than 0.1. There is another way to do this with the shorthand. So instead of making this mask variable, you can just like put that whole thing in these square brackets here. And that is the exact same thing as doing this DF breast cancer with the mask here. Great, okay. So I think that's the longest case study of all of them because I think it's probably the most important. So we're gonna move on to exploration, grouping, and visualization. So we're gonna want to look at this healthcare insurance costs data set. So this is insurance.csv, and we are just gonna get right into it, read it in. So we have done this pd.readcsv. I have determined beforehand that there's no need to add any other sort of arguments in there to make sure it's quite a well-behaved data set in that way. It just seemed to work fine the first time. So it looks like we have a few attributes and then this charges feature. So based on age, sex, BMI, the number of children, whether they're a smoker, and where they live, there is potential correlations between those and how much they've claimed in insurance. So this is an interesting data set. So first, we're going to want to take a look at what the data set looks like. So I like this little df.describe. It gives you a pretty easy high-level description of each of the attributes. Unfortunately, without any arguments, it just gives you the numeric ones, because with numeric data you can give the different quartiles and the mean and standard deviation, where you can't really do that with like categorical. I do like including the categorical, so if you add this include is equal to all, it will give you everything. So, you know, for the categorical variables, all of these will be not available, but it will tell you how many unique categorical variables there are, the most common category, and then also the frequency of the most common category. And so in like, say, smoker, that's the same thing, in region, you know, what's great is that using count, you can figure out if you have any missing values. Lucky for us, there don't seem to be any missing values. But definitely there's another way to figure out if there's missing values. So you can use this .isNull, which will check if every single value is null or not. Then you tack on this any, and then on a column basis, it will tell you if there are any null values, and it looks like there are not. And if you tack on one more any, it will tell you if any of the columns have any missing values. And so if this is false, then you're good to go. There's no missing values. So let's do some data exploration, I guess, just to kind of see if we can immediately see any patterns. So what we want to do is group by. So if anybody has taken a databases class, this should be a very familiar concept to them. So let's first do a group by sex. So we have the data frame, we have group by, we give it the attribute to group on, and then we ask for the counts. So in this case, we've grouped by sex, so then each of the values in this categorical feature will be given a row, and then based on how many of these rows appear to have values in each of these features, it will return that. And so because there's no missing values, all of these will be the same across here, and all of these will be the same across here. But that's just an easy way to figure out like how many males and females there are in this data set. You can group by multiple variables if that's interesting to you. So say you want to see the breakdown of the sexes in different regions. You could then specify a list, sex and region, and then this count as well. And so then for females, you can see which regions seem to be like the most common, and for males, which regions seem to be the most common. And then for, if you're interested in, say, like a numerical column, you can use sum. So in this case, I'm going to say df insurance.group by sex, and then take the sum. And really the sum of the age and the sum of the BMI and the sum of the children is maybe not so interesting, maybe the children a little bit more so. But if we want to specify charges, for instance, now we have figured out the sum of all insurance claims like filed by either sex. So that's an interesting exploratory tool. So visualization is, I have here something that deserves its own workshop, and I can't give you too much of it, and I'm not going to explain exactly how all of this code works, but I would absolutely say that if you are interested in visualizations, look at this link. It gives a great overview for how to do all these top 50 types of visualizations. So first of all, I'm going to show how to check the distributions of the data that you have. So say we're interested in the distribution of BMI. So here's a very simple way to plot a histogram. This is using matplotlib that I imported earlier. So the value of interest is a list of all of the BMIs of everybody in the insurance data set, the data frame. We specify here the number of bins, and then we use this plot.hist, and then we specify the values that we have and the number of bins we want. Here is just making the plot a little bit nicer with labels, but I'm going to run this. And there it is. It shows the distribution of BMIs. And frankly, this data looks very well behaved. This looks like a pretty good approximation of a normal distribution. The skew doesn't look too bad. So I would say probably not a lot of treatment is necessary for this particular attribute, but it's possible that you will have very skewed data in the future. So looking at that is important. And if you wanted to do a transformation, so say you saw the distribution and you thought this needs, like this is so skewed, this needs like a log transformation. A great way to get these kind of transformations is to use NumPy, which is this NP. And then here we have a log base 2, and we do the log base 2 of this column, this BMI column, and then we can create a new column in our data frame by using this kind of like shorthand. This only works if this attribute does not already exist. And I do think that's pretty confusing. But anyway, if you wanted to do a log transformation, this is how you would do it. So I run that, did the log transformation. So now within this, we should have BMI and log BMI if we wanted it. Okay. So now another one I would like to go through is the plotting categorical variables. This is also using matplotlib. So let's say we want to see what this group by, this GB, looks like. So I'm going to run it. This is the group by of whether people are smokers or non-smokers. I've just included age here. It's not a big deal, just to make it a series rather than a data frame. You could have chosen any of the remaining attributes. So what we're going to want to do then, and I think this workshop might be a little bit longer than an hour now, but because you can skip through, I'm not going to sweat about it too much. So here we have the labels. So in this case, gb.index is this no and yes. And then the counts, this gb.tolist are these numbers here. So now we use plot.bar, which gives a bar plot. There are other types of plots that you can do, which I'll show you in just a second, and then adding the labels. And so here we have a nice looking plot that shows whether people are smokers or non-smokers in the data set. So now by replacing bar with other words, like off the top of my head, I know scatter is one of them. You can get something like this, which, you know, in my opinion is not the most effective way to show off how the data is, but a lot of these require similar setup. So it's easy to switch between different types if that's what you're interested in. Okay, and let's get to case study 3. This is the one I'm most excited about, and this is actually something that I've worked with and published research on. So we're going to be using the Cancer Genome Atlas. So what we're going to want to do here is deal with missing values, because in the real world, data has lots of missing values, and we're also going to be joining data sets here. Okay, so we have two data sets here. We have this TCGA genetic data, and we have the TCGA clinical features. So first, we're going to want to load in clinical features. So I have this here, index column equals 0, because otherwise we were going to have the same problem as we had before. So This data has over 11,000 rows and has 33 columns. If we look through the columns, it shows quite a bit of like demographic information as well as information about this person's cancer and vital status, days to last contact, new tumor event sites, that kind of thing. Today we're not going to be dealing with all of this information because it's just kind of too much, but I would like to just show you what a real data set actually looks like, rather than what I showed you before, which is just kind of like a nice cleaned version of the data that is good for learning like data science techniques. So what we're going to do is we're going to define the columns we want to keep for this analysis. So I would like to keep patient barcode because that's going to be our join key. We want overall survival time. We want age at initial pathologic diagnosis. We want the residual tumor, and we want the tumor stage. So what we do is we assign this data frame to the columns that we want to keep. And then this is our final DF clinical that we have now. Okay, so let's do a describe where we include all of them. And this will give us a good idea of maybe what kind of transformations we're going to need to apply. So first of all, patient barcode. It looks like, if we look at the number of rows versus the count, it looks like every single person has a barcode, which is fantastic. That's what we need because joining is important. Slightly fewer people seem to have overall survival time. A few people are missing their age when they were initially diagnosed with cancer. It seems that a shocking even amount of people do not have this attribute. So if it's NAN, then this number is going to be, I guess if they have a value, they will be included in this. So it looks like only around 10% of people have any value at all for this column. And then we have this pathologic tumor stage where we have about maybe like 75% of the people have this attribute. Okay, so let's go column by column and figure out what we're going to need to do to the data for this. So let's imagine that we want to predict the overall survival time. So this is very relevant to what I do. Because that's our target attribute, if a row does not have a value for it, we need to get rid of it because it doesn't make sense to include them in our machine learning training, because how are we going to predict what they have? And that's not going to do any good for the model. So what we're going to do is we're going to get rid of all of the rows that do not have a value in this row or in this column. So I won't go over this too much, but basically we want the data frame such that the attribute overall survival time is not NaN or not null. So this keeps all of the non-null values, and if we do this, and if we want to take a look, DF clinical. If we want to take a look now, we have excluded a few of the columns that did not have the value for overall, excluded rows that did not have the value for overall survival time. Okay, so I'll delete that. So now we move on to age at initial pathologic diagnosis. This is how old the patient was at the time of their diagnosis. And there are a few NANs here. It's pretty good. So what we're going to do is we're going to impute the values. So we are going to replace empty cells with something that is, like I said before, unoffensive or innocuous, like the median value. So if the value is basically the median for any row, then it's unlikely that that attribute is going to contribute a lot to the final decision that the model is going to make, like say, what their final survival time is. So it's, especially when there's not a lot of missing values, imputing based on the median is a great way of just, making the problem of missing values go away. So what we're going to do, we're going to get this variable existing median where we take the median of this entire column. Then what we're going to do is we're going to select the column with this age at initial pathologic diagnosis, use the function here fill null, so fill the null values. We're going to fill it with the existing median, and we're going to say in place equals true, meaning that we don't have to set this whole thing as it. So this, instead of it just performing it on a copy of the data frame, it's going to do it to the data frame itself. So we might get an error. Yeah, that's a bug. Just ignore it. Pandas and these these Python libraries are not perfect. They're maintained by like a very small number of people who just do this out of the goodness of their hearts. So this is fine to ignore. So yeah, now we should not have any missing values in our age at initial pathologic diagnosis. Okay, so if we look back up at residual tumor, this value and this value are very different. And if we have that many missing values, it's probably the best bet to just get rid of it. So what I do here is I say dfClinical is equal to dfClinical.drop, and then I feed in the labels residual tumor, and then I say the column. I specify that this is a column, not a row, by saying x is equal to 1. And then there, I have dropped it because it's probably not going to be that useful to us because there's so many missing values. And then the last row that we want to deal with is this pathologic tumor stage. So if we look up here, yeah, so they actually give the number of tumor or the stages of these tumors for different types of cancer. And so because this is a string, this is categorical, we need to do one-hot encoding like I had mentioned before. So first we want to fill null values, and I use the in place is equal to true, because we want to make it so the rows that have a null value are treated as their own class. So we're pretending that not having a value is its own class, its own value of that column. And that allows the model to use that information. Perhaps that's even relevant. So we're going to do this in place. So we're modifying the actual dfClinical variable. So now we have filled the null values with the string null value. And if we do this group by here, we will be able to see the different classes. So there's quite a few different stages, and as you can see now, null value, which there's about 2,200 or so, this is now treated as its own category, and this is a really wonderful way of dealing with null values in categorical data. So we have all of this, and now I'll show you how to get the one-hot encodings. So there's this get dummies function. And all you have to do is feed in the column that you want to get the one-hot encoding of. So this is just a series, but dummy df becomes an actual data frame. So this has the same number of rows as before, but a different number of columns. So here you can see, like, for stage 4, it has 1 because this second row is stage 4. So anyway, that's great. And because it has the same number of rows, you can just kind of like take the previous data frame and the new data frame and just stick them together because the axes line up. So what we're going to do from dfClinical is we're going to get rid of our original column, which was, we're going to get rid of this column here with the actual categorical stage information. And we are going to stick the dfClinical and the dummy df together, and we're going to create the final data set. So here's what everything looks like. So this is where we're at. So we have patient barcode, survival time, age at initial pathologic diagnosis, and then we have velcroed all of our one-hot encoded dummy variables to this. So now we have 25 columns and still a decent number of rows. And let's check for null values, and it looks like we have done a great job of getting rid of all of the null values. So excluding this patient barcode, because this is just something that we're using to refer to rows, everything else is now pretty close to ready for machine learning. Okay, so now, yeah, this ended up being kind of a longer tutorial than I was thinking. So I'm going to keep going, skip parts that you don't think are interesting if that's relevant for you. So let's quickly load in genetic data. So now this might take a little bit. So this is Illumina mRNA-seq data. And so each value represents the number of, or like the pseudo number of transcripts that existed at the time of measuring. So this is measuring gene expression. So these columns do, so here, the number is the Entrée ID. There's a vertical bar separating it, and then if the gene has a gene symbol, it will be on this side. So this is how the data kind of looks coming out of, if you do mRNA-seq. Obviously, some pre-processing probably has to be done. But in this case, we have the clinical barcode, which matches up with this clinical barcode in the clinical data set. We also have the biopsy barcode because actually people can appear in the biopsy genetic data set multiple times if they have multiple tumors. In this case, I have selected only 500 of the probably 10,000 genetic rows that we have here, just because otherwise it would just be, the data set would be too big, it wouldn't fit on GitHub, etc. So this is what we have to work with here. So one way to deal with null values, instead of getting rid of the rows, we can get rid of the columns. And in this type of genetic data, often the null values are, they imply that the measurement for that gene or for that column or feature is poor kind of all the way through. So what we're going to want to do is we have 20,533, or I guess 31 genes, and we're going to want to get rid of all of the ones that have null values. This will make our problem go away. And so we're going to do this df-mRNA drop nan axis 1. So drop any columns that have a null value in it. So looks like that was pretty quick. And like I say here, we lose about 4,000 genes, but often this having these values means the gene measurement was poor anyway. And you can see like a lot of these values are 0 anyway, probably not that interesting for machine learning. So let's take a look at what it looks like now. So we have about 16,000 and some genes, but lots of really beautiful non-null values here. So this is what we wanted. Okay, so let's combine these two data sets. I'm going to use pd.merge. So this is analogous to the join that I was talking about earlier. I have chosen the inner join because we only are interested in patients that have both clinical information and then by extension the survival time information, as well as patients that have genetic information, say if we wanted to predict survival time based on their gene expression. So I'm going to do this merge here. So this merge has, the first two arguments are the two data frames that you want to merge. You specify on with the column name, so they should have some column name in common. And we say how is equal to inner because, like I said before, we only want patients with clinical and genetic information. So we'll do the merge, and there we go. So we have now 16,361 columns. We have all of this clinical information, and we additionally have their genetic information. So, and then here's the biopsy barcode if you wanted to have that, and the patient clinical barcode here. And so this is our final data set. So let's look at shape. We lost 2 of the 500 genetic rows because they didn't have associated clinical information, so we didn't want them. This is definitely smaller than the number of clinical features, but again, that was because of the inner join. And then before machine learning, we don't really care about the patient barcode anymore, so because we have all the information in the right rows anyway. So we are going to drop the barcode and biopsy code. Okay, so there we go. Then if we wanted to look at DF merged, this is what the data set finally looks like. So, okay, this is a lot of information. I'm definitely over time, but I'm going to keep going very quickly. A lot of this stuff here is review. So if you want more detail, go back and look at my other workshop. But because the data frame containing the breast cancer information is so nice already, this is what we're going to use to demonstrate preparing data for machine learning input. So we already have this read in from earlier. Check, looks good. Everything looks normal. We have the target here that we're trying to predict. So what we're going to do is we are going to create our X and Y data. So X contains all of the attributes that we want to use to predict Y. Y contains what we want to predict. This has been covered, I think, multiple times. We are going to use this train test split function that we imported earlier, probably I think from a scikit-learn or something like that. This will give us the train and test X and Y's here. Here we don't need a validation set because we're not doing any parameter tuning, and so that will be, maybe talked about sometime in the future. Then let's just run these so we have everything. Yeah, so this is what X looks like, this is what Y looks like, and now we have X train, X test, Y train, Y test. So this is just kind of a manual way to scale the data. So this just makes sure, once I run this, that every value for every column will be between 0 and 1. This makes the machine learning step quite a bit easier. So we're going to run that. That's just so the weights that the data or the model has to learn can all be like kind of in the same order of magnitude. So let's take a look at what this looks like. So X-train, it sampled a bunch of the different data points, and now everything is between 0 and 1, which is very, it's important. It can never hurt in my experience. So then let's look at, well, this is just a duplication, but it's fine. So anyway, we have the shape here, 455 and 30, 455 rows, 30 columns. If we look at the X test, we have 114 rows and 30 columns. And then when we look at the Y train and Y test, this is the Y train here. This is the Y test here. And so these are the train ground truth and the test ground truth. So that's it. That is the preparation that we've done now for machine learning. So here I have next steps. This is where you come in. I think that now that we have all this great data, this is a wonderful place to add your own exploration. Add your own potential machine learning model on top of it to train it. Now that we have a lot of the data already pre-processed and ready to go, if you need a hint, use the X train scaled and Y train to train the model, and then the X test scaled and Y test to evaluate the model. And then, like I mentioned many times, if you would like a more in-depth example of a machine learning model, in my Google COLAB workshop, I use support vector machines to do that. And then all I have to say is have a wonderful evening. So I will leave this on my my contact card. If you have any questions at all, please reach out. I'm happy to chat. This is like my favorite thing to do. So here's my email. I have a LinkedIn as well if you would like to connect, and a Twitter that I don't use that much, but sometimes I post like funny stuff. And then for everything AIMS related, check out linkter.e/AIMS. and that will have links to just about anything that you would need. All right. Thank you so much for sticking through it. We're at about an hour and 10 minutes, so we're a bit over time again, but it's a little bit more flexible this time. So I hope whatever time of day it is for you, you have a wonderful rest of your day or a week or evening or weekend or whatever. And that's everything from me. Thank you so much for sticking around and watching and really being excited to learn. Thank you.